All right, now to finish off chapter 6. This is obviously a long chapter, so let's talk about it. Okay, so carrier-mediated transport. So, okay. Okay, so the phospholipid bilayer is semi-permeable. Okay, molecules that are large or polar or charged, okay, cannot diffuse across the membrane. Okay, examples: amino acids, glucose, other organic molecules. Okay, that's why there's so many embedded proteins in the plasma membrane that move these things across. Okay, the characteristics, these are specific to a given molecule. Okay, that there may be competition for similar carriers or other molecules. There is saturation, so there's only so many, that hole can only fit so many things through at once. And you know, the more carriers you have, the faster you can get things through. Okay. All right. Some proteins can transport more than one molecule, like similarly charged things, but there's competition effect. Okay. Slideshow. The transport rates, okay, not rats, increase with increased molecule concentration until saturation is met. So there's a maximum, there's a transport maximum when all the carriers are used. Okay, and this is the same. This is like the same graph that you guys saw with enzymes. There's actually a maximum. So at this point right here, okay, you are pumping in whatever X is as fast as you can. Okay, if you have less carriers, then this is as fast as you can go. Okay. So facilitated diffusion. Facilitated means that there's no ATP. Diffusion means that there's no ATP used. Facilitated means that you need a carrier. Okay. All right. Net movement from high to low concentration requires specific carrier-mediated proteins. Transport proteins may exist in the plasma membrane or might be inserted when needed. A good example of this is aquaporins. Okay, so we insert them when we need to pump water through. Okay, so here's the diffusion of glucose, facilitated diffusion of glucose. Okay. All right. Transport carriers for glucose are designated GLUT, followed by the number, which is the isoform. GLUT1, CNS, GLUT2, etc., etc. Okay, GLUT4, adipose and skeletal muscles. Okay, this can be inserted into the plasma membrane of skeletal muscle when stimulated. This is why exercise, okay, actually helps diabetics. When you build up muscle, when you build up skeletal muscle, it increases GLUT4. Increased levels of GLUT4 allows the glucose to be absorbed into the blood, from the bloodstream into the cell without insulin. Okay, most cells require insulin for glucose absorption. The GLUT transporters do not. They suck up insulin they suck up glucose regardless of whether insulin is there. Okay. Okay. So here, you make a bunch of carrier proteins, okay, in the ER, and then when they get transported, okay, to the surface, okay, they move through the plasma membrane. Again, this is stimulated by either insulin or exercise, okay, but the exercise actually causes these transporters to get there even without insulin. Okay, active transport. Molecules can move from low concentration to high concentration, okay? But this always requires expenditure of ATP. Okay. So primary active transport, okay, occurs only when hydrolysis ATP is directly responsible for the carrier protein function. Okay, so the transport protein is also ATPase, which means it uses ATP. Okay, so usually you phosphorylate the ATP, and that causes a conformational change, which allows one thing to move across, and then you have to burn enough ATP to do this over and over and over again. Okay, nice. All right, so the calcium pump. The calcium pump, okay, we are moving from, okay, so we're using ATP, okay, so we're pumping things out of the cell here, okay, and we're burning ATP in the process, okay. This is a particularly important pump, okay, the sodium potassium pump. It is found in all body cells. The ATPase enzyme pumps 3 sodium out of the cell for 2 potassiums to enter the cell. That is a very big thing. Now I've set up a condition where all the sodium is going to be on outside and all the potassium is going to be on inside. That's one thing. So now there's a chemical imbalance across the cell membrane. Okay. The second part is now there's a charge imbalance on either side of the cell membrane. I pumped out 3 positive charges for 2 positive ones. So there's going to be a net negative charge inside the cell. So now I've set up a charge difference across the membrane. Okay, so I set up a electrochemical gradient across the membrane. Okay, so I provide energy for coupled transport of other molecules, which we'll talk about shortly. Okay, I just mentioned it produces electrochemical impulses in neurons and muscle cells, and what maintains the osmolality of the cell. Yeah, so here it is. Okay, every time this ATP goes, I trade 3 sodiums, okay, for 2 potassiums. Okay, so secondary active transport is also called coupled transport. Okay, okay, so let's go through an example of this. Okay, now I have all my sodium stuck outside the cell. That sodium naturally wants to come back through the membrane into the cell. Now I can hijack it for something. Okay, so I can use it to bring glucose in. The sodium wants to come in, I use it to bring a glucose along with it. Okay, so secondary active transport of glucose. So this part, I am not using any ATP here, so you don't see any ATP here, but I use ATP to get the sodium out of the cell in the first place. So it actually costs me energy for secondary active transport of glucose. Okay, I can also counterport it where I move something in the opposite direction. Okay, so one sodium comes in that wants to come in, and in response to that, I pump out a calcium. Okay. All right, transports across the epithelial membrane, absorption, reabsorption, okay. Okay. Absorption is the transport of digestive products across the intestinal epithelium, okay. Reabsorption is reabsorbing what you filtered out in the filtrate. When you're in the process of making urine, you make something called filtrate, and then you reabsorb everything you want back in. Okay. All right. Okay. So, okay. So here. Okay. So this is the kidney tubule or the small intestine. Okay. You can bring in glucose here. Okay. And then you purposely pump out glucose here. So you keep, this is facilitated diffusion. So glucose keeps leaking back into this bloodstream. So you can then bring in more glucose this way. Okay. So the transportation of sodium down this concentration gradient. Okay. So down this concentration gradient. Okay. I keep this purposely low, right? So now this is inside the cell, so I keep the sodium purposely low here by using ATP, and now I can get the glucose. Okay. Okay. Junctional complexes. Okay. Let me just quickly go through what these are for. These actually hold the cells together. It needs to hold the cells together and prevent tearing. The tight junctions do something slightly different. See how they're higher on the cell surface here? They actually prevent things from leaking between the cells. Okay. Okay. Bulk transport. Okay. Endocytosis. Let's talk about this. Okay. So endocytosis has 3 steps. One is the invagination of the plasma membrane. Second is the closure of the loop here, the formation of the pouch, and then this breaks off, and now you have a vesicle. That vesicle can now be moved inside of other stuff. Okay, like for example, a lysosome and stuff. Okay, this is the exocytosis is the direct opposite. So I make things from the rough ER. From rough ER, it goes to the Golgi apparatus. From the Golgi apparatus, it fuses into the plasma membrane. Okay. Okay. There is a difference in charge on each side of the plasma membrane due to the permeability of membrane. Okay. The action of the sodium potassium pump, and naturally there are big molecules that are negatively charged inside the cell and they can't exit. Like for example, the DNA is negatively charged. So there's going to be a charge difference. Okay, inside of the cell is going to be negatively charged compared to outside. Okay. Okay. All right, so I will constantly pump out plus charges because the electrical attraction is to constantly bring plus charges in, and so we're constantly pumping those out. Okay. Okay, the membrane potential, potassium. Potassium accumulates at very high concentrations inside the cell because one, we're actively bringing in the potassium. Okay. 2, remember there's more negative charges inside the cell, and potassium is actually permeable. okay, and allows it to come through. Okay. All right. And so the negative ions inside the cell attract positive charges, and this one can come in easier. So, okay, it's limited by strong concentration gradient. If you bring in too much, it'll start to leak out. Okay, so the concentration inside the cell, okay, is 150 EQ per liter. Outside is 5 EQ per liter. Okay, milli EQ. Okay. So the potential difference can be measured as voltage. Okay, so the voltage. Just based on potassium should be negative 90, okay, but not actually negative 90. Okay. Okay. The other thing is sodium. Okay. Sodium is the opposite. Sodium is kept on the outside much higher than on the inside. Okay. So there you go. There's the intracellular concentrations. Okay. Versus the extracellular ones. These three are the ones that really drive the charge difference, the voltage difference. The calcium, not so much. Okay. Okay, so the Nernst equation is used to calculate equilibrium potentials. Okay, it's based on the concentration of ions. Okay, so here's the equation. Honestly, I don't think you have to use it, so I don't get into it too much. Okay, but here's the Nernst equation for sodium and potassium, and you can see they go in opposite directions here. Okay, there you go. The membrane potential almost skipped forward to membrane potential. So in most living cells, the resting potential is between 65 millivolts and between negative 5 millivolts. Neurons are usually around 70 millivolts. This becomes really important in the next chapter when we talk about how neurons work. Okay. Okay. So here, there is a negative charge, negative voltage of 70 millivolts inside the cell. Okay. Okay, so the role of the sodium potassium pump, we mentioned this already. It acts to counter this, the potassium leaking out. Okay, so it transports in, it maintains the voltage difference. I already explained that earlier. It keeps both the resting potential, okay, at negative 70, and the concentration difference is stable. Okay. Okay. So again, the resting membrane potential is negative 70. Okay, and now I have the other thing I want to point out is the uneven distribution of ions across the plasma membrane. Okay, so I have potassium leaking out. I have sodium on the outside dying to get in. I have, in general, positive charges trying to get in because there's more negative charges on the inside. Okay, and then we will pick up this thing next chapter. Okay, when we talk about membrane potentials and how neurons actually work. Okay. A little break. All right. Sorry about that. So All right. So cells communicate using chemical signals. So gap junctions are small gaps in between cells where the signal can travel from one cell to another. Okay. Paracrine, para, around. Okay, it's like endocrine signaling, but not without the bloodstream. So they dump it directly to the outside, and it just goes to the cells that are around them. Okay. Okay, synaptic signaling, neurons, endocrine signal, bloodstreams. Okay, so blah blah blah. Okay. Okay. For a cell to receive a signal, it has receptor protein specific to it on the plasma membrane or in the inside the cell, especially on the nuclear membrane. Okay. Okay. Nonpolar molecules, steroid hormones, okay, can go through the plasma membrane directly, so they can get into the receptors on the inside of the cell or on the nuclear membrane. Okay. Large polar molecules, they have to interact only on the plasma membrane. So the receptors have to be on the plasma membrane. Okay. Okay. So the large signals bind on receptors on the cell surface. So from the cell surface, you need to go into the cell interior, and those are the cyclic messengers. Okay. So secondary messengers, okay, are going to be small little things like calcium or cyclic AMP, but they have to be small and they have to be able to move quickly through the cytoplasm of the cell. Okay. So blah blah. Okay. So if the regulator is polar, then they have to use the second messenger. If the regulator is nonpolar, they can actually go directly into the cell and interact with the receptor, either in cytoplasm or on the surface of the nucleus. Okay. Okay, cyclic AMP. Okay. So CCAP, cyclic AMP, okay, is a really common second messenger. The steps to activate. So a signal molecule binds to a receptor. This activates an enzyme that produces cyclic AMP from ATP. Cyclic AMP is then goes through the cytoplasm and activates other enzymes. And cell activity change response to those other enzymes. Okay. Okay. Receptor proteins. Okay, so here. Okay. Okay. You have the regulatory molecule, you have the receptor, and then you have the G protein here. Okay. When the regulatory, usually these are not actually this close. Okay. When the regulatory molecule binds to the receptor, what happens, okay, is that the G protein becomes activated. Okay, it exchanges at GTP, it cleaves at UTP, and therefore it becomes activated. Okay, now these two now separate, and these guys can have different effects on different effectors. The alpha subunit and the beta subunit can have different effects on different receptor effectors. Okay, Usually the alpha subunit works on the surface, and then the beta goes activates other things, IP3, etc. That works differently. Okay, but eventually the signal goes away, and they reattach, and they go back to square one and wait for a new signal. Okay. All right. Okay. Hopefully that will.